Alrighty, hello everyone. My name is Jack Butler, and today for my long talk, I'm going to be talking to you about tau misprocessing in a blast-exposed mouse model that develops CTE neuropathological characteristics. So before I get real into detail, I want to give you a basic outline that I'm going to be going through throughout this long talk. Firstly, I'm going to give you a diagram of the overall pathway, I like to call it, of brain injury to what happens after that, and then how does that lead to neurodegenerative diseases. It's just really a broad overview that's just supposed to help you understand it a little more throughout this long talk. Then I'm going to go into background information, then the research question kind of slash hypothesis of this paper, the overview of tau, giving you a schematic diagram of tau, how it's associated with neurodegenerative diseases, and what can be consequences of tau. Then I'm going to showcase you images of tau misprocessing. Then I'm going to take you right into the experimental approach that these researchers took by using a mouse model to develop their data. Then I'm going to talk to you about the analysis of the data, following up with conclusion and wrap up, which is essentially just what's next and really the overarching theme, I should say, of this long talk. So here is the diagram of the overall pathway, I like to call it. So what we have is impact, which is what I have in parentheses is TBI, which is traumatic brain injury, which is defined as damage to the brain caused by impacts such as a bolt or a jolt to the head. Most notably, you might think of concussions in football. This is a great example of traumatic brain injuries that can result from getting hit really hard in the head. Another one that is important is blast exposure. While this may or may not follow the same mechanism, it also can induce this traumatic brain injury. So what happens after this traumatic brain injury is tau misprocessing, which I'm going to explain more in following slides, which then ultimately leads to neurodegenerative diseases such as CTE, which is chronic traumatic encephalopathy, which is a form of neurodegenerative disease that causes severe and irreparable brain damage as a result of repeated head injuries. Also, Alzheimer's disease, which is just a type of dementia that affects memory, thinking, and behavior. There's also Parkinson's disease. That's another neurodegenerative disease. Some symptoms that are associated with these diseases. We have dementia, which is memory loss, confusion, erratic behavior, difficulty with motor skills, confusion, trouble staying focused. So another thing that I want to point out here is this question mark that is between impact and tau processing. And this is something that we don't know, which is how is tau becoming misprocessed? We don't know this question. But regardless, what the researchers are trying to find is not to answer this question, but rather a different question. So some background information. What do we know? Well, we know that traumatic brain injuries, TBIs, which just told you about in the last slide, have been linked as a precursor for neurodegenerative diseases such as Alzheimer's disease and chronic traumatic encephalopathy. This was just on the previous slide that I just showed you, which is just the broad overall pathway from head injury to tau misprocessing to neurodegenerative disease. Next bullet point is microtubule associated protein tau can be hyperphosphorylated and has been connected as a pathological indicator for development of neurodegenerative diseases. I'm going to give you a better overview of the tau protein and how it can lead to neurodegenerative disease in the following slides, but just know that this tau protein is really important for neurodegenerative diseases. And lastly, blast exposure can induce characteristics of CTE neuropathology in both animals and humans. This is important because for the researchers, they can use this information to then compare their mouse model to humans using a blast exposure device, which they create. But this is important because their results are going to be similar in the fact that blast exposure can induce this characteristics of CTE neuropathology. So getting into the research question, what are they looking at for here? So I told you that they're not trying to find the mechanism of hyperphosphorylation, but rather they're looking at the difference between concussive injuries and blast exposure. And if these two mechanisms of developing neurodegenerative diseases share a common pathway. So they're looking at, do they share a biomechanical and pathophysiological process between the two that ultimately leads to the development of CTE. And so the hypothesis that is formed from this is that if they share a common biomechanical and pathophysiological process, then they will showcase similar indicators of CTE neuropathology. So yeah. So again, just they're looking at the mechanism here. Is there something in common between these two? So now for tau, I'm giving you the schematic of tau protein here. I'm not expecting you to know too much about neurons. I guess the only thing I would like for you to know is that neurons, they send signals throughout your brain in response to certain functions throughout your body, to motor functions, basic memory, learning, talking even, all these basic functions. These neurons are consisted of different parts in their entire kind of composed cell. But what important part in respect to what I'm going to be talking about is this axon, which is stabilized by microtubules. Well, these microtubules are then further stabilized by tau. So this picture illustrates it really well. We have the blue dots, which is the microtubules. The orange lines are tau. So what can happen, and this is a question that we still don't know, is these tau can be hyperphosphorylated. What this means is that phosphate groups can attach to these tau proteins in large numbers. So there can be multiple phosphate groups on these. And what can happen as a result of this is that these tau proteins will disassociate from the microtubule and become aggregated together to form these clumps. And what these clumps are called are NFTs, or neurofibrillary tangles. And that's what we can see here. Over here on the left, we can see the clumping of these proteins, and they form these tangles. And then what happens with the microtubule is it becomes like untangled. It starts to disintegrate. And ultimately, this leads to axonal defects and neuronal death. So tau protein is really important because you want healthy neurons. And if you don't have healthy neurons, then you're going to have neuronal death, which is a cause for neurodegenerative diseases. So here's another good schematic diagram showcasing you the difference between a healthy neuron and diseased neuron and the impact of tau. So what we can see in the healthy neuron is the microtubule being stabilized by this tau molecule. And then in the diseased neuron, we see that the tau protein is forming these clumps. We can see the disintegrating microtubule leading to non-stabilization of this axon, and it ultimately leads to neuronal death. The diseased neuron clearly looks a bit more tattered and weared than the healthy neuron, which is not good. And this is causation for neurodegenerative diseases. So what do all these tau clumpings look like in brains? Well, this is what they look like. So in the first two rows, A, B, C, D, E, F, G, H, I want you to look at these brown spots. So in the first row, we see the brown miscoloring, which is tissue death. And if we zoom in on this miscolorization, we start to see these brown/black dots. What these dots are those NFTs, those neurofibrillary tangles, which are, again, those hyperphosphorylated tau all clumped together. What we can see in comparison to that is healthy brain. So at the bottom row, A, B, C, we see that there is no discolorization, no brown tissue. We also don't see any little brown dots. So these NFTs are clear markers, and we are able to see these in brains. And these cases of brains are taken from not only military veterans, but also from young athletes who have recorded concussions and have later died as a consequence of these. But the takeaway from this slide is that I want you to be able to see what these NFTs look like in brains. So now jumping into the experimental approach, the mouse model that they used, how were they able to recapitulate this blast that US military veterans are exposed to in a lab? So what they did here is they used compressed gas. And what this gas did, or this compressed gas, was it would drive the shock tube that was used to deliver a highly reproducible sublethal blast shock that was relevant to a human blast injury. So this mice here in this picture that is being used is placed in this prone position, is kind of half of its body's kind of hunkered down while its neck portion up is being able to freely rotate, which is similar to how, football players would be, or in this more particular case, how military veterans would be in respect to having a blast exposure. Their head is going to be moving around a lot while they're, being blasted. Another important thing is that this shows, or this model that they use, is really comparable to what a US military veteran or US military soldier would experience in the field. This IED blast that US military soldiers experience, which is just a improved experimental device, would be with roughly like 4.5 to 5 kilograms of C4 at about 5 meters. And what this mechanism or device that these researchers were able to craft is that it showed nearly identical in respect to the amount of C4 and the distance. While this model is not actually using C4, it is recapturing that same kind of like disaster that it would occur. So the main takeaway from this is that this mouse model was used to recapitulate an IED blast that was commonly encountered by US military. So let's get into the data of this mouse. So what we see here is in these 4 panels taken from separate brains of the mouse is that there is a difference. And so one thing that I forgot to mention in the last slide is that the mouse were introduced into a single blast or a sham blast, which was called a control blast. And so this is what M&R is showing is the control blast or sham blast. NNS is the single induced blast. So in M&R, we don't really see too much discolorization as in respect to NNS, where in N and S, we see this brown color existing, which is, dead tissue. And then in S, we really start to see these brown/black dots, which again are those NFTs. So what this tells us is that single blast induces the CTE-like neuropathology in these mice. This is a rather important slide, and I think really cool slide, because it's showcasing you that not only what they are seeing is correct, but on a more analytical test that their results are also correct. So what we are seeing is just a bunch of gels, and within these gels, we see different colored bands. Some might be lighter than the other, some might be darker. And this is what this is showing us is levels of protein concentration, in particular tau. What it is showing is darker protein or darker band color means more protein concentration of tau. And so you might be asking yourself, why are there 3 different, panels? And this is showcasing you what I like to think of it as is within a protein molecule, we know that it's, made up of many amino acids. Well, what if we wanted to just look at in particular 1 or 2 amino acids within this total protein? And that's what 2 of these are looking at. So going through just kind of the figures and legends of this, we see that all the ones on their left are the control blasts or sham blasts. All the ones on the right here, blast 1 and 2, these are the blast exposed mice. What the left and right means is hemisphere of brain. It's just, 2 sections taken from different halves of the brain. What the 53 kilodaltons means is this is the molecular weight of the protein, the phosphorylated tau protein. And since we see that it is the same throughout all these different gels, this means that we are looking at, in fact, the same protein. So this is good for us. And now let's get into the kind of the wording up here. So we have CP13, PS202, PT205, AT270. What does this mean? This CP13 and AT270, it's really kind of just like a common name to describe the specific amino acid that you would be looking at in a protein. So here it is serine 202. So serine at position 202 and 309 at position 205. Down here, we have 309 at position 181. What this means for us is that, and I guess what is rather important about these positions is that they are associated with early neurodegenerative tau misprocessing. So the key word in that is early. So that's probably why they looked at this, is because they wanted to, get the results back in a quickly, timely manner. And so they looked at these specific amino acids in the tau protein to generate the results. And so in E and F, it's tau 5, which is, again, just kind of like a common name, but this is for total tau. So now we're not just looking at a single part in the amino acid or in the protein, rather we're looking at the whole entire thing. And so what we can see in all of these results is that in the sham blast or control blast, the bands are a lighter color. In the blast exposed mice, we see that the bands are a darker color. This means that the protein concentration of hyperphosphorylated tau is increased in blast exposed mice. And this is really important because not only did we just like see the images in the brains in the last slide, but now we're seeing analytical data, and that we're seeing that, okay, the concentration of this hyperphosphorylated tau is increasing in these blast exposed mice. So now that we have kind of this, color device showcasing us increased hyperphosphorylated tau, another thing that they did is now they quantified this. So now they put this in numerical terms, and we're able to show that this hyperphosphorylated tau in the blast exposed mice is significant. And so this is what the next slide is showcasing. So here I say quantification of phosphorylated tau shows the increase of p-tau, phosphorylated tau, in single blast mice. So what we have on the axis here, we have the sham, which is controlled. We have the blast, which is the single blast exposed mice. And then we have the two single, amino acids that we're looking at in the entire protein. CP13, it was the serine at position 202 and the 309 at position 205. And then AT270, just kind of a common name for the 309 at position 181. And so again, these positions are known as early, or are as known as being associated with early neurodegenerative tau misprocessing. And what we can see, or I guess I'm getting a little ahead of myself, and then on the y-axis, we see phosphorylated tau in arbitrary units. So we don't necessarily have like a unit for it, and that's why it's in arbitrary units. But regardless, what we see here is that in both of the, different specific regions within this whole protein that we're looking at, we see that there is more phosphorylated tau in the blast exposed mice. And both of these are significant, indicated by the little stars here. This one has a 0.005 significance, and this one is 0.001. So clearly more phosphorylated tau is being seen in these blast exposed mice. And this is just a quantification of what we just saw on the last slide to determine to us like, okay, we're putting this in numbers. We're able to get a numerical value that, hey, this information is significant. So the big takeaway from really both of these last two slides is, again, that phosphorylated tau is increased in blast exposed mice. So now what we might be looking at is, okay, what is another way that we can show that concussive injuries and blast exposure share, similar biological pathways? Well, what we might do is let's try to point out what might prevent these, neuropathological characteristics that lead to CTE. And so that's what they did here. So the basis of this experiment that they kind of did here was, like I said in the previous mouse model, that they let the head rotate free. Well, what they did here was they immobilized the head. So they kind of put up guards so that the head, couldn't move when being exposed to this, compressed gas. And so what they looked at is, rather than analyzing the, rather than analyzing portions of the brain and looking for, those NFTs within the brain samples, they rather looked at the symptoms being associated with neurodegenerative diseases. So what we talked about in the beginning, dementia, trouble focusing, erratic behavior, loss of motor control, all these kind of symptoms, this is what they looked at in this kind of sub experiment that they did. So what these two figures are showcasing is they immobilized mouse in the blast chamber, which is indicated by the blue color. They then did again blast head free, which is just, there was nothing. The head was allowed to freely rotate. And then they did the sham blast, which is their control. They then put these mice in mazes and they allowed them to go try to find the exit. And what they looked at is, on figure D, over a 4-day span, how long that mice took for them to find the exit. And what we can see is that the blue and the black one, so the blast head fix and the control blast, took roughly about, the same time over the corresponding days. Whereas in comparison with the head free, we see that mice took longer. So what does this tell us is that these mice with the head free are, developing symptoms such as, confusion, memory loss, motor functions that would, allow them to escape the maze slower. Whereas the blast head fixed, they are not necessarily developing these symptoms because they are able to escape the maze at roughly the same amount of time that the control blast mice are escaping. And this is the same thing kind of for B. This is just kind of showcasing, or in E, I should say, rather, as like a bar graph. So again, it's showcasing black as control blast, red as head free, and blue as head fix, which is showing that the head was placed fixed. This is showcasing, how long the mouse was able to escape. And what we can see is that the difference between head free and sham was significant, and head fixed and sham were close to the same. There was not a significant difference between the two. So this can tell us that symptoms associated with neurodegenerative diseases are causing the head free to lag in escaping this maze. Whenever you immobilize the head, they are not developing these symptoms, meaning that they're not developing these neurodegenerative diseases nearly as bad as the head free. So what all of this tells us is that, hey, since, we're immobilizing the head, maybe it's that this rapid acceleration, deacceleration of the head is the cause for neurodegenerative diseases. And so that's what they take away here. So the conclusion, indistinguishable CTE-linked neuropathology was observed in both blast exposure and concussive injured postmortem brains. The blast mice revealed really similar evidence of the CTE-linked neuropathology. The mouse model developed symptoms that were really indicative of neurodegenerative diseases. And lastly, this is the main takeaway, the research question that they were asking, is that they found that this bobblehead effect, quick acceleration, deacceleration of the head, was shown to be a common injury mechanism between CTE cases. So really the wrap up of this is why does this matter? Well, it's important because if football players who are getting concussions and U.S. military veterans who are developing PTSD and other symptoms associated with the brain are, dying to the same reason of CTE and Alzheimer's and other neurodegenerative diseases, well, now we know that they might share a common pathway. So now we might be able to treat that the same way. We might be able to do prevention methods the same way in respect for both of them. And this is really important because we have a lot of U.S. military veterans that are out there serving for our country. And there's also a good amount of football players who are dealing with concussive injuries that ultimately will lead to neurodegenerative diseases. So if we can find a common, mechanism which was found in this paper, we can find maybe a common prevention method and effective treatment. So I hope that this paper, or not this paper, but this talk taught you a lot about tau in the brain, leading to neurodegenerative diseases, what is shared between blast exposure and concussive injured athletes, and that this ultimately can lead to neurodegenerative diseases such as chronic traumatic encephalopathy and Alzheimer's. So here are my references, and thank you guys for listening.